Thank you very much. Good afternoon. It's a pleasure to be here, back from Boston, and I just moved there, as you heard, and plan to stay there a few years, but I don't rule out coming back maybe later on to Europe again. So it's great to be here. Great presentations this morning, and I would now discuss in 30 minutes as much as we can on the biology of pancreatic cancer. And actually, when you give this talk today, the good news is that there is a lot more that have been discovered and a lot more that we know about this disease compared to a few years ago. The bad news is that none of, or very few of these discoveries are being incorporated into clinical care. So I envision that the next few years will be quite interesting and exciting in incorporating part of this biology in the way we manage patients. Needless to say that we are facing one of the most important cancers with regards to mortality. You know that recently was announced that is now number 3rd in cancer-related death, causes of cancer-related death, and soon to be probably number 2nd, only behind lung cancer. And that gives you an idea of being probably number 10 in frequency, how high the mortality of this tumor is. And this is a graphic showing these trends with regards to incidence and survival. So significant problem and a problem to stay in the next few years. Now another disappointment is that when you look at what happened in clinical cancer and translational cancer research over the last few years, we are starting to see that in many diseases that a few years ago were non-treatable, now precision medicine and immunotherapy is inching up the survival of these patients and making an impact. While pancreas cancer remains one of those in which neither one of those new strategies is so far paying results with regards to patient outcome. And I would talk about these two areas and what new discoveries are out there and what we think could be put forward in the clinic in the near future. And this is just an outline of what I said before. We manage these patients with chemotherapy, either for FOLFIRINOX or gemcitabine Abraxane in the first line, and then a number of other medications in the second line, but really no impact or no significant impact in the outcome of the disease. It's true that patients live longer, but we still have a long way to go. So to start discussing what are news or exciting new developments in the biology of pancreas cancer, I would like to talk about 3 particular areas. Talk a little bit about the cancer cell per se, the mature cancer cell, and what we have learned about its genomics and how can we start exploiting this. Then talk about the cancer stem cell as a group of cells that may be or have been considered to be important in cell renewal and are resistant to chemotherapy and radiation therapy, and maybe a particular group of cells of interest to target, and then talk about the stroma and immunology and see where we are in that particular area. The good news, as I said, is that finally many of those discoveries being done in the preclinical, more basic laboratories are starting to be translated into clinical research. And this is a summary of what I think is now a very exciting field of new agents being developed in phase 1, phase 2 studies in pancreas cancer. Now, all of them look quite promising, all of them look quite attractive, but we know that many of them look the same way and then they failed in the clinic. We were talking in the break today that it's only March and we already learned about the failure of Threshold, failure of Alpine, the Notch inhibitor, and more recently the JAK kinase inhibitors as well. So 3 new drugs put forward, all of them failed. Many more to come, we'll see what happens with them, but certainly exciting in the sense that there are many new strategies. Now, the first look at the genetics of pancreas cancer was done back in the 2006, 2007. This was work that was done initially at Johns Hopkins. I was part of that group that sequenced the first few genomes in this disease. This was actually done before Next Generation and Illumina and all that was available. So this was all Sanger sequencing of 20,000 genes per case. So as you can imagine, a lot of work, a lot of PCR, but the picture that it provided, I think, is quite similar to what we have learned later on. And this is a disease that is characterized by having a significant genetic complexity, as you can see, with a mean or median of 60 aberrations per patient. Significant heterogeneity. There are not two patients that are the same, so there are patients that have, there are some mutations, we will talk about then, that occur in the majority of patients, but then there is a myriad of mutations that occur in very small groups of patients, and of course, this will be kind of the holy grail of precision medicine if we were able to identify which one of those are important and develop drugs against them. And part of the problem, which is that all this sequencing effort at the end led to this conclusion that most of these tumors have mutations in one of these 4 genes, KRAS, P53, SMAD4, or P16. And unfortunately, at the moment, we don't have effective drugs, effective strategies to target neither one of those. So it's, I think, unlikely that the field of precision medicine will have a significant impact unless medications to combat some of these genes are available. Because again, most of the patients, if not all, will have 2 or 3 mutations in these genes. Now, there may be some new drugs that are now attacking those particular alterations. Just to give you an example of some work that we did at the preclinical level with a pan-HER inhibitor, in which we treated models that have, these are patient-derived models, that have KRAS mutations, all of them, but the difference between these two groups is that these are P53 wild type and this is our P53 mutated. And here we saw very clearly that this agent, which is a pan-HER inhibitor, targets the ERB1, ERB2, and ERB3, are effective in blocking the growth of the P53 wild type, not the P53 mutant. So this is being advanced to the clinic with this particular subset, KRAS mutant, P53 wild type. This may be up to 20% of the patients, and we will see what the results are. More sophisticated genomic analysis have been conducted mainly by Andrew Bianchi and the Australian group as part of the international TCGA and International Genomics Consortium, and have come out with a more perfect, if you want, genetic landscape of pancreas cancer, more complete, more than perfect. And out of this work that was recently published, what they were able is to classify these patients based on whole genome sequencing in 4 groups. And at least some of these groups, like for example, the unstable group, which is characterized by alterations in DNA damage repair genes, could be patients that are candidates for treatment with PARP inhibitors, alkylating agents, or drugs that target this particular genotype. Now, the problem with this approach is that we are not ready to implement this in the clinic. We don't have the technology in our hospitals that a patient comes in, you take a sample, a few days later comes back saying you are unstable, because this requires, is still very complicated to come out with this conclusion. So how you apply this to the clinic remains, I think, a significant challenge, and actually a challenge or a boundary that we'll need to cross if we want to take advantage of this data available. It's true that when you identify a patient, individual patient, with a single mutation in some of these pathways, then you can treat that patient with drugs that are supposedly effective against it, and you get sometimes good results. And this is a patient that we treated a few years ago with mitomycin C based on the observation that A, had a mutation in the PALB2 gene, the only one that we found in the 24 patients with this mutation. B, that this mutation was important, in other words, was functional, impair DNA damage repair, and see that a model developed from this patient was susceptible to alkylating agents. So this was the rationale to treat the patient with these drugs, mitomycin and platinum, and the patient actually did very well for many years. So there may be cases out there where this information can be already translated, but these are the minority. There have been, again, some other efforts to classify pancreas cancer. This is work by Eric Collison and colleagues, utilizing, in this case, gene expression profiling. And what you see is that you can cluster patients in different groups, and these groups have different prognosis. So they came out with three classes of tumors, 3 groups, and as you can see here, the quasi-mesenchymal group has a worse prognosis. Another ramification of this work is that there is evidence that some of these groups may respond differently to the drugs we already have in the clinic. And here you can see comparing two of these groups with regards to the activity of gemcitabine in here and erlotinib, and you see that there are groups that respond better than the others. Again, this is interesting, may have a mechanistic rationale, but how to classify patients in these groups is still something that we don't have available. Actually, another problem is that those classifiers are not, in general, very robust. They have been done with, let's say, 100 patients. But if you add 5 more patients, they will be classified in one group. But when you redo the same experiment, now considering 105 patients, because you added 5 more, sometimes the classification changes. So this is still complicated if we want to do it sort of in a clinical approach, because of course it will need to be robust to be utilized in the clinic. You cannot be changing the group that the patients belong from one day to the other. More subgroups. This is also from the Bianchi's group, recently published in Nature, utilizing also genetics and then gene expression profiling, and they came out with 4 groups: squamous, pancreatic progenitor, immunogenic, and aberrantly differentiated exocrine, endocrine neoplasia. Same concept, different prognosis. Of course, it's very exciting if the immunogenic group is the group that will respond to immuno-oncology. This is something that remains to be studied. But again, how we use this methodology to classify our patients in the clinic is still to be developed. This is the project that we're still doing in Madrid, in Senayo. This is going to stay there. I moved, but the project will stay, in which we're trying to kind of incorporate all that knowledge and query, when we have patients, try to analyze as much as we can their genomics. This will help us to come out with potential treatment strategies. Simultaneously, we do an avatar model of that patient, test in the avatar what have been obtained by this work, with the idea that some of these will go back to the patient on one side, and on the other side that we will be perfectioning these databases as we incorporate more clinically validated genomic outcome correlation. So this is a large project, just started. Actually, I think I picked the wrong time to move, because I decided to go, and then this was funded, but again will remain at Senayo and will be done as a collaboration in a few hospitals in the Madrid area. Has been quite complicated to get this off the ground. I could not appreciate, when we were doing this, the regulatory complexities. This is a prospective clinical trial in which you are randomizing patients to do either standard of care or all these integrated genomics, where you put DNA on one side and you don't know what is going to be coming out on the other side. And the regulatory agencies will say, well, you need to tell me what is going to be coming out on the other side, whereas I don't know. Well, that doesn't work. Then list all the... So it's been quite an interesting few months dialoguing with them. Finally, we got it approved, so it will start running. So let's continue moving down the number of things that I wanted to tell you today. Let's talk now about cancer stem cell. Just to give you an example, it is known that this subgroup of cells, among many other characteristics, have very distinct signaling pathways. And some of them are important because are more targetable than the pathways that occurred in the mature cell. Just as an example, it is believed that these cells have alteration in the notching pathway. And indeed, work that we have done in collaboration with Oncomed indeed have showed that when you block some of these signaling pathways with a notch inhibitor, you start getting tumors that respond very well to treatment, and importantly and differently from chemotherapy, you get tumors, and these are PDX models, that stay under control for a significant period of time, suggesting that they may indeed have been cured, and this is associated with a significant decrement in the content of cancer stem cells that these models have. So based on these observations, we started a clinical trial with a notch inhibitor, an inhibitor of the delta ligand 4. As part of this project, we started a co-clinical trial approach in which the patient goes into the trial and receives the medication, but the avatar model that we routinely establish from some of the patients in clinical studies is treated with the same drug, and therefore, at the end of the day, we can compare outcomes. And what was interesting in this study is that we saw some patients, like this patient, that have been treated with the inhibitor of Notch and did not do very well, indeed progress very rapidly. This was done in combination with gemcitabine. But when we combine gemcitabine with Abraxane and the DLL4 inhibitor, we started to see that the triple combination was clearly more effective than the doublet, listed here in blue, much more effective. So this was interesting kind of data that was generated at the same time the clinical trial was being conducted, that prompted, of course, the amendment of the protocol to start adding Abraxane, and since we did this, we have seen quite significant response in many of these patients. So this drug is now being tested in a randomized phase 2 study called the Yosemite, which many centers in the world are participating. Certainly very exciting preliminary data, but again, we will need to wait for the results because we have seen some of those before that then in the phase 3 didn't pan out. So let's go just to the final few minutes to talk about stroma and immunology. All this story about the stroma, I think, started by the initial paper by Ken Olive and Dave Tubeson back in 2008, where they started to show that something that has been known before, that those tumors are very rich in stroma, but they started to think on ways to block or eliminate the stroma, with the idea that the tumors that are, in general, pancreas cancer, very hypovascular, when you eliminate the stroma, perhaps you could increase vascularization, and by that means increase chemotherapy delivery. And indeed, working with a hedgehog inhibitor, they were able, in preclinical models, to show that if you eliminated or if you inhibited the hedgehog pathway that was predominantly activated in the stroma, you will increase drug delivery and by that reason, efficacy. And this was very exciting at that time and led to clinical studies. Actually, I flipped the slides here, but unfortunately, the first generation of studies done with hedgehog inhibitors and gemcitabine were not that impressive. Actually, if you look at the median survival here, you would say it's short. So this kind of was a little bit of a setback in the field, but some other studies, however, have not been so disappointing. This is combination of IP926 with FOLFIRINOX, where you see that there is a significant number of patients who had a response. However, because the field is kind of already, or was already considered not to be effective, many of these drugs have been discontinued. But the interest to target the stroma persists. I'll give you a couple of other examples, and then we'll go back to the hedgehog inhibitors. This is work that we did in parallel to the clinical development of nab-paclitaxel, in which we showed that there was a significant activity of this drug in preclinical models, in PDX models, and actually that was one of the reasons why the drug was kind of rallied to clinical development and in 4 or 5 years was essentially approved. That was, I think, a very nice story on how working together you can make sometimes these studies go faster. But part of the observation that we made initially is that this drug here eliminates the stroma. And this was of interest because, of course, will open up the possibility of increasing drug delivery or doing other combinations. Not all groups have been able to show this data. So I think the effects of Abraxane in the stroma is still kind of a moving target. But we saw this initially. We have seen these also in more recent experiments that are now submitted for publication that importantly showed that paclitaxel doesn't do the same thing. Very clearly shown here. Stroma intact, stroma depleted, intact, depleted. So we're trying to understand whether or not Abraxane is still a drug that targets the stroma and the implication that this may have. We went ahead and did also a study in the preoperative setting in which we treated patients for a couple of months with gemcitabine Abraxane and then collected their tumors. And once again, we were able to see that the nab-paclitaxel treated group had a very, not eliminated, but very altered stroma. And what the consequences of these are remains to be seen. But certainly this is one of the agents that we could consider targets the stroma. And the most recent one, which the one that is more advanced, is pegylated hyaluronidase, the PH20, which based on preclinical studies done by Tubeson and Hingorani, they propose that it will eliminate hyaluronic acid and by that mechanism increase basal patency and increase chemotherapy delivery. And as you know, this is in clinical trials at the moment. Again, in the preclinical models, the combination of this agent with gemcitabine was more effective than the gemcitabine alone, so positive preclinical data. And in what has been already presented with regards to the clinical data, if you look at this row here, the hyaluronidase high patients in this study, the progression-free survival is quite substantially improved. So this is one of the agents, and I think we need to keep an eye what happens in the future. So at least 3, maybe more, drugs, Abraxane, hedgehog, pegylated hyaluronidase that target the stroma, some of them positive data, some other very positive early data, some other negative data. So what is going on? Well, I think that preclinical studies are starting to show some light into that. And these 2 studies from 2 different groups showed, in essence, that the acute elimination of the stroma, mainly by hedgehog inhibitors, may have positive effects with regards to drug delivery, but the chronic elimination of that stroma leads to a situation where the cancer increases proliferation, and because of the increased angiogenesis, increases also metastasis. So the benefit that you may be getting early on is lost as the treatment continues, and these were shown in some of the preclinical studies in which, indeed, administration of a hedgehog inhibitor was detrimental, which is very different from the first study published by the same group. And the difference is indeed in the timing. So this may explain why the studies were negative, may suggest how we redo these studies if we were to redo them. But it also pointed to a very interesting observation, which is that while the tumor becomes more aggressive, one of the things that is observed is that the inflammatory or immune infiltrate increases. And by doing that, of course, they ask the question, well, will a checkpoint inhibitor work in these circumstances? And the answer is yes. They got very interesting preliminary data with ipilimumab combined with depletion of SMA-positive fibroblasts. So it really opens the possibility that these stroma-mediated agents may be a mechanism by which IO will be effective or could be effective in pancreas cancer. And of course, there is a significant interest in understanding why pancreas cancer is so immunosuppressive and does not respond to immune oncology type of approaches. And there are a variety of strategies or pathways that have been proposed. Some of them are already prime for clinical time. For example, the notion that CXCR4 ligands are responsible for keeping lymphocytes out of the pancreas cancer stroma, and I will show you some data in these regards. So it's well known that one of the characteristics of pancreas cancer is that they have very little lymphocytes. And of course, that's a problem. If there are no lymphocytes, it's very unlikely that a lymphocyte activating drug, like maybe a checkpoint inhibitor, will work. Well, what has been shown is that when in some of these preclinical studies, treating tumors with elements that depleting FAP positive cells, or treating tumors with CXCR4 inhibitors really increases the number of lymphocytes in the tumor and permits synergy with immunotherapy. And this has been shown by genetic means, and more importantly, has been shown also by pharmacological means, using a CXCR4 inhibitor in combination with anti-PD-L1. So there is a lot of interest in moving this group here, this combination, to the clinic, and in fact, there are a few studies that are already ongoing combining these two strategies in patients with refractory disease. So these mechanisms, together with the mechanism of eliminating the stroma, may be pathways to get immunotherapy into pancreas cancer at the end. And finally, just 2 slides to show another, I think, very recent and interesting paper in which investigators engineer T cells to recognize, in this case, mesothelin, some of the more specific pancreas cancer antigens. And at least in the preclinical models, they have shown that this strategy of T cell receptor engineer cells, similar to a CAR T cell but a different mechanism, is able to improve the survival of otherwise very hard to treat mouse models of pancreas cancer. So I think that based on all these mechanisms, there's going to be a wave of studies combining checkpoint inhibitors with other drugs that may modulate the stroma or the immune suppressive tumor microenvironment, and also some cell-based adoptive treatments that either alone or in combination with other agents likely will be developed. So as I said, very exciting new opportunities for pancreas cancer. And just to conclude, of course, this remains a significant, very important health issue. Unfortunately, we still manage patients with surgery, radiation therapy, sometimes, and chemotherapy, with the results that are not optimal. Precision medicine, I gave you a few examples. IO, I gave you a few examples, still early days, but I think based on the better biological knowledge, these treatments will eventually come to the clinic and hopefully show efficacy. And just let me finish by acknowledging the different contributors to this work. Thank you.